world is facing an environmental crisis of epic proportions, and one of the largest contributing factors to this issue is the way we receive our energy. While they power the homes of millions, fossil fuels have become the necessary evil of the 21st century. Now, a solution to all the problems that energy poses is biofuel, or fuel derived from renewable biomass material. Now, while there are various ways of making biofuels, every single process has proved counterintuitive. However, making biofuels would take up twice the amount of carbon dioxide present in the atmosphere than current efforts, because the process of growing and harvesting crops for biofuel uses CO2 as a fuel source that, among its many other environmental benefits, it should definitely not be wiped off the table. In comes algae, a simple non-flowering plant that includes the micro and macrovarieties. They do not have true stems, roots, leaves, or vascular tissue. And inside the endoplasmic reticulum of each algal cell lies lipids and several other important facts. Now, while there are many different types of algae, the research that I conducted focused on three specific types: spirulina, which is often studied for its high lipid count in the form of omega-3s; Microalgal, which is often studied for its rapid expansion and inflation rate; and Microalgal, which has been studied for biomass in the past for similar reasons as chlorella. While algae has been overlooked for many decades, the promise of utilizing the lipids inside algae cells is currently being investigated. In essence, the algae would have to grow in photobioreactors where lipids would have to be separated from the plant media. However, this would require both a large budget and a willingness to use a large amount of energy, and no single optimal process has been determined by any energy company. Thus, I set out to find a process for myself. I grew algae with the intention of harvesting biofuels. I altered conditions such as the light and waters that I used to see how that would affect the amount of biomass produced. And when it came time to harvest biofuel from the biomass that I grew, I altered a ratio of ethanol to biomass to extract my biofuel. And this is my independent variable. My dependent variable was the amount of biofuel extracted at the end of the day. I hypothesized that because algae grows under natural conditions, it would be most optimal for algae to grow under natural light. And because of the fact that natural water often has hindering contaminants in it, I hypothesized that algae grown under clean water would do the best. So for the first week, all the culture was placed in natural light just to bring it into the live phase, and we're then divided in half with water from the Mill Creek River and half with clean, sterilized water. Afterwards, I divided the culture in half again to ensure that my results were verifiable. And I logged my observations continuously throughout that process. So about 2 weeks into the initial procedure, I began to notice a thin layer of biomass in some of the algal flasks, and from then on out, I saw an exponential growth of algal biofuel, and the first cultures to bloom were indeed those grown with clean water and natural light. So based on my charts, it is seemingly clear that spirulina must be the one that produces the most biomass. However, at a quick look at the nanochlorolysis, you can see that it does phenomenally well in contaminated water. Now, this poses the question, will it do good in an energy crisis? And the answer is yes. If we needed to use it in natural bodies of water in any nation, it is possible to do so. Then came the process of extracting the oil. I used vodka containing 40 to 50% ethanol, and I put it in a small frying pan with the biomass. After much trial and error, I discovered that a 2-to-1 ratio of ethanol in the form of vodka to biomass worked perfectly by setting the pan on high and then lowering the temperature throughout a 40-minute process. Once I began to notice that the biomass brewed in a bubbly fashion, the biomass eventually removed itself from biofuel, and that was my end product. Conclusively, it is indeed possible to extract lipid biofuel from 3 genus of algae. The sources in which the algae receives its water and light do matter in optimizing the production of biomass for biofuel. The photosynthetic process provided by natural light is beneficial to algal culture, and the hindering contaminants in water make it clear that clean, sterilized water would be best for this process. The experiment also allowed me to create my own extraction process in which the ethanol and vodka stimulated a chemical reaction with the algae in order to extract the lipids. The Microalgal, which was the most healthy, biomass-rich species, not only produced the most oil, but proved to be the most effective. Biofuel is indeed the most promising and economical secure energy. However, it is still a work in progress as biochemists, bioengineers, and other innovators begin to create new methods for successful high-scale production of microalgal biomass and the implementation of this new technology for algal biorefineries in the future. Thank you.